Swayam Prabha. Digital India. Educated India.
Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the generation of genetically modified organism, and in the scheme of genetically modified organism, what we have discussed so far, we have discussed about the different host strains, their physiology, the biochemical reactions, what they catalyze, and what is the relevance of these biochemical reactions for the organism as well as for the industrial significance. And in continuation to that discussion, we have also discussed about how to isolate a gene of interest from the particular organism, so that you can use the gene fragment into the downstream cloning reactions and to generate the genetically modified organisms. So, what we have discussed so far is that if you have a genome, and from which if you use the polymerase reactions to generate the gene, you can also get this fragment by the either by the genomic libraries or the 3DNA libraries. So, whatever the sources, after that you have to digest these gene with the restriction enzymes, and what will happen if you do it the restriction enzyme, you are going to generate the cohesive ends on both the sides, and the similar kind of procedure you have to do for the transforming agents such as the plasmids, and then you have to put it for the ligation, and then you will get the ligated plasmids. These ligated plasmid you have to put it into the host strain, and that is how you are going to get the transformed bacteria or the genetically modified organisms. During this whole procedure of generating a genetically modified organism, you have the multiple enzymes which are being used in this complete scheme. And in the today's lecture, what we are going to do is we are going to discuss about the different enzymes what we use in the cloning reactions, and their mechanism, as well as how to perform or how to use these enzymes in the lab as well. So, as you can see that we have the 4 major enzyme. One is the polymerase. This polymerase enzyme we have discussed in our previous lecture. The second enzyme is called as the restriction enzyme or restriction enzymes. These restriction enzymes are also being required to generate the cohesive ends, and the third enzyme is the enzyme which is required for ligating the your insert as well as the plasmid DNA. So, in totality what you have is you have the 4 major enzyme which are having the applications in the molecular cloning. The first class is called as the restriction enzymes. The purpose of the restriction enzyme is to cut the DNA at a specific site. This DNA could be the plasmid or could be the your gene of interest what you have isolated after the PCR. Then you have the polymerases. In the previous lecture we have discussed about the PCR amplification utilizing the polymerases which are belonging to the tag DNA polymerases. Then we have the alkaline phosphatases. The purpose of alkaline phosphatases is to remove the terminal phosphate group, and that also has the specific applications in the molecular cloning. and then the last enzyme is called as the DNA ligase. The purpose of DNA ligase is to join the two DNA strand. In this case, one could be the plasmid, the other one could be your insert which you have digested with the restriction enzyme. So, let us start with the restriction enzymes first. So, the restriction enzyme is a part of a big system which is called as the restriction methylase system, and restriction methylase system is like a immune system of the bacteria or the lower organism. So, as you know that what is the function of the immune system in the higher organisms? The purpose of the immune system of the higher organism is to not allow the external organisms to grow or to proliferate. That is being done by simply having the different types of barriers. For example, in a human being, you have a skin which actually protects your body from getting the entry of bacteria, fungus and all other kind of infectious organism. Below to this skin, you have the first layer of defense which is being mediated by the macrophages or other kind of immune cells, and after that you have the real immune cells which are like B cell and T cell and all those kind of T lymphocytes and B lymphocytes, and all these lymphocytes are having a similar function that they are not going to allow this particular infectious organism to grow into your back cell. And what is the advantage of having this system? Because it will not allow the opportunistic infection or the other kind of infectious organism to draw the nutrition from the host. Similarly, the bacteria as well as the smaller organisms have the restriction methylase system to not allow the propagation of the particular DNA of the external sources. So, how this restriction methylase system works is that every restriction enzyme, for example, in this case we have taken an example of EcoR1, has a specific site which is called as GGAATTC. So, it is having a specific site, means a specific sequence of nucleotide, and that would be cleaved by the EcoR1, but under the two conditions. One, either the DNA will be unmethylated or the DNA will be the methylated. If the DNA is unmethylated, which means that this particular DNA is not going to belong to that particular organism, and if the system will see a unmethylated DNA, the EcoR1 will going to cleave this DNA and will generate the sticky ends. So, by doing though, it is actually going to generate the external DNA into the DNA fragments. But suppose this particular DNA is belonging to the EcoR1 or the E. coli itself. In those cases, what will happen is that the EcoR1 methylase enzyme, which is actually in the also the part of the restriction methylase system, is going to methylate the specific site onto the restriction site. So, in that case what will happen is that if this DNA belongs to the E. coli itself, then what in the E. coli what will happen is that the within this recognition sequence, the crucial adenine is going to be get methylated by the enzyme, and because of this methylation group, what will happen is that the restriction enzyme EcoR1 will not be able to bind the DNA and will not be able to cleave, and once it is not done, then it is not going to degrade the DNA, which means that restriction methylase system also has the specificity, it also has the ability to recognize what is self and what is non-self. For example, in this case you might have seen that this particular DNA is considered as the non-self simply because it does not have the methylation on the adenine groups, whereas the DNA containing the same restriction site but methylated on the adenine group is considered to be the self DNA, and that is actually a unique mechanism to distinguish between the self DNA versus the non self DNA. Now, how the enzyme will going to know which one is self and which is non-self is because the restriction methylase system has the 3 different activities associated within the single enzyme. One is called S activity, the other one is called as the methylase activity, and the third one is called as the restriction methylase activity. How that happens? That you suppose, imagine that you have a piece of DNA with a 4 restriction sites RE1, RE2, RE3 and RE4. Now, you have the 4 sites and you have added the restriction enzyme or restriction methylase system, and which actually contains the 3 activities, the S activity, R activity and M activity. The S activity is activity which actually going to recognize the specific sequence onto the DNA, and it will then lodge the restriction enzyme onto the DNA of your interest. So, what will happen is this enzyme is going to interact with the RE1, RE2, RE3 and RE4, and all these 4 sequences are different. So, that is why the S activity will going to interact, and while it is interacting, it either will form the open complex or the closed complex. Once it is binding, it may have the very high affinity for the RE2, because this is the site which is actually been recognized by the S activity within the restriction enzyme. So, once it is going to recognize, it will go and bind to the RE1. Now, you can imagine that the enzyme has been launched onto the DNA with the help of the S activity. Once the S activity will be launched, then it will insert its M activity into the enzyme to detect or to check whether the DNA is methylated or hemimethylated or the unmethylated. If the DNA is unmethylated, if DNA is methylated, the M activity will find these methyl groups, and then it will not going to give the access to the restriction site, restriction activity to come into the picture. But if the DNA is hemimethylated, which means that if you have the site and one of the site is methylated, the other strand is non-methylated, in that case what will happen is that the M activity onto this strand is going to insert, whereas on this side the M activity is going to generate the methylation. So, if the DNA is hemimethylated, the restriction enzyme system, restriction methylase system will say that okay, this is the DNA which is being generated from the parent DNA, and it is actually been our self DNA, and because of that what will happen is that restriction methylase system is going to add the methyl group onto the next strand, just so that it will be get immunes from the getting the cutting of DNA. But you can imagine that if you have the restriction site and it is non-methylated, so S activity is being used to provide the site where the DNA is going to bind by the enzyme. The M activity will detect whether the methylation is there or not. If there is no methylation, then M is also get inserted into the DNA, and then the R activity or the restriction activity will come and find and bind and form the closed complex. And once it is formed the closed complex, which means the R activity can actually be able to access the DNA, then it is actually going to cut the DNA into the 2 parts, and these 2 parts are going to be released. Once these 2 DNA fragments are going to be released, the enzyme is also going to be released into the solution, and it will again be ready for the next cycle of catalysis. So, restriction methylase system is a very precise system adopted from the microorganism or the smaller organisms to protect themselves from the propagation of bacteria or viral DNA or other kind of DNA from the other organisms. Restriction methylase system is also going to distinguish, this is what we have already discussed, between self and the foreign DNA, simply because the foreign DNA is going to be unmethylated compared to the self DNA, which is either going to be methylated or the hemimethylated. So, if the same site is present into the hemimethylated form or the or methylated form, it is considered to be the self DNA. If it is the same site which is present as the unmethylated form, then it is considered to be the foreign DNA, and that is what the way the restriction methylase system is distinguished between the self or the non-self DNA. Now, since the restriction enzymes are being discovered, there is a classical or there is a particular rule which people are following to write the name of these restriction enzymes. So, if you can see an example, what you can see in an example, you see the example of the EcoR1. So, EcoR1 is one of the enzyme. Okay. So, what you are so the nomenclature is that the first letter you will take as the genus of that particular organism. The second is the species, whereas, so in this case EcoR1, E is E. coli or Escherichia coli. So, E is Escherichia, the CO is coming from the coli, and then R is actually the information about the strain. Okay. So, for example, in this case you have the R strain, and the fourth letter is the order in which the enzyme has been isolated from the particular organism, which means that if you have isolated the EcoR1 the first time, this is going to be the first enzyme from the E. coli strain R. And so, that is how the people have started naming the different restriction enzymes. For example, you have the BAMH1, you have HIN3, you have HE3, you have XHO1 and so on. So, all these restriction enzymes have a critical pattern in which their name is being put, and so that actually helps in getting the information about the enzyme. It is also getting the information about the source from which you have isolated the enzyme. There are different types of enzymes, different type of restriction enzymes. So, restriction enzymes are vary in their structures, recognition site and the cutting site and the cofactor requirement. Based on these parameters, the restriction enzymes are of 3 different types. One is called as the type 1 restriction enzyme. The type 1 restriction enzymes are the recognition site is consist of 3 to 4 nucleotide at the 3' end, followed by a non-specific stretch of 6 to 8 nuclei and the 4 nucleotide at the 5', which means the type 1 restriction enzyme recognition site is going to be a very long and non-specific, and then this is the recognition site. Once it is recognizing, the cleavage site is approximately 100 base pair away from the recognition site, which means that the enzyme is actually recognizing here and binding here, and whereas it is cutting the DNA away, which means the type 1 restriction enzymes are first of all have a very uncharacterized or loose recognition sites, and then where they will cut is actually the 1000 base pair away from the actual recognition sites. They also require the cofactors. One is called as the S-adenyl methionine, ATP and magnesium for their activity, and the type 1 restriction enzymes has the dual enzymatic activity, which means they can be able to do restriction as well as the methylation, and the enzyme which and that is because the enzyme has the 3 subunit, one is which is called as HSDR, HSDM and HSDS, whereas where HSDR is for the restriction, M is for the methylation and S is for the recognizing the particular DNA sequence. So, apart from that, you have the type 3 restriction enzyme. The type 3 restriction enzymes are having the restriction site which has the 2 separate non-palindromic sequences arranged inversely oriented. The cutting site, so cutting site in this case is 20 to 30 base pair away from the main recognition site, and the type 3 restriction site is also composed of 2 subunit. It is restriction site as well as the modification site. Mod activity or the mod subunit is required for the modification, which means the methylation of the DNA, whereas res subunit is required to cut the unmethylated DNA. So, whether it is the type 1 site or the type 3 site, both of these restriction enzymes are recognizing at particular sequence, but they are cutting at a different site or the cutting at a very distant site. In the case of type 1 enzyme, it is actually cutting at 1000 base pair, whereas in the case of type 3, it is cutting approximately 20 to 30 base pair away from the recognition sites. Now, the third is the type 2 restriction enzymes. So, type 2 restriction site is the recognition site is 4 to 8 nucleotide long, and it cuts the DNA within the restriction site, which means that if you have this DNA and the enzyme is recognizing this particular stretch of DNA, it cuts the restriction site within that particular site. Due to this particular unique feature, the type 2 restriction sites have an application in the molecular cloning, because you will need an enzyme which actually precisely cuts where it actually goes and bind, and you should know that what sequence it cuts and what sequence it binds. Whereas, if you see that in the type 1 and type 3, they were binding to somewhere and cutting to somewhere. So, that is why you cannot predict or you cannot generate you cannot use those enzymes for the molecular cloning. It is composed of 3 subunits, which is called M, R and S. So, similar to the type 1 enzyme, the M, R and S have the similar kind of functions. M is going to do the methylation or the modifications, R is for the restriction, and S is for the recognition of the particular DNA sequence. The type 2 enzymes are also of diversified functions. So, they are also been classified further into due to their unique feature of each class. So, in the type 2E, these class of enzyme contain the DNA which actually contains the 2 cleavage site, which means if you have the 1 cleavage site, these type 2E enzymes will not going to cut the DNA, whereas if you have the 2 sites, the first site is actually being required to induce the activation of the enzyme, and then the enzyme actually cuts onto the DNA on the other side. Similarly, you have the type 2B, and these enzymes actually are cutting the both sides of the recognition site to remove the recognition sites from the DNA, which means that if you have the restriction site like this, it actually cuts on both the sides, and that is how it actually removes that fragment of that particular DNA. Third is the type 2M. The type 2M is the resection in the sites which actually cuts the methylated DNA. So, the contrary to other restriction methylase system, these enzymes are actually cutting the DNA when it is methylated. So, one of the classical example is DPN1, and the DPN1 has a very classical usage because of this unique property that it actually cuts the methylated DNA. You might have as we discussed, like methylated DNA is immune from getting the cut from the restriction methylase system, but in this particular class of enzymes, the enzyme cuts the DNA because it is methylated. And what is the application of this particular enzyme? This particular enzyme is always being used to detect or destroy the host gene when you are actually generating the mutations. How you use this enzyme is that suppose you are generating a site directed mutant into a particular type of wild type gene. In those cases, what will happen is that you use the primers to mutate the site. Okay. So, in that case, what will happen is that you have taken a plasmid and you did the PCR with the mutated primers. In those cases, what will happen is that you are going to get the 2 plasmids, one which is going to be the wild type, the other one is going to be the wild type, and the other one is going to be the mutant. Once you got these 2 kind of species, you will transform these particular reactions into the host, and so both of these species are capable enough to grow into the host, and will give you the colonies. So, what is the difference between the wild type and the mutant is that the mutant is non-methylated, whereas the wild type is methylated. So, in these kind of situation, what the people do is they treat this reaction after the PCR. You got these two plasmids, and what they do is they just treat this reaction with the DPN1, and what will happen is the DPN1 is going to cleaved or will going to destroy the wild type enzyme, whereas keeping your mutated plasmid intact. And then if you transform that into the host, you are going to get the colonies, and that colony is going to contain only the plasmid for the mutant, and then you can take out the colonies and do the sequencing to confirm that the mutation is being done. So, that is what is the application of these restriction enzymes which are belonging to the type 2M. Now, let us see what is the differences between the different restriction enzymes. So, you have the type 1, type 2 and type 3. As far as the recognition site is concerned, the type 1 is non-palindromic. Okay, we will discuss about what is palindromic and what is non-palindromic. Type 2 is palindromic and type 3 is also the non-palindromic. The cutting site as we discussed is 1000 base pair away from the recognition sites, whereas in the case of type 2 it is within the recognition sites, and in the type 3 it is 20 to 25 base pair away from the recognition sites. The enzyme composition, in the case of type 1 you have the 3 subunits, one is called HSDR, HSDM and HSDS, and all these individual subunits have their exclusive functions, whereas in the case of type 2 you have the R, M and S subunits, whereas in the type 3 you have the 2 subunit, one is called RES, the other one is called as the MET. Cofactor, in the case of type 1 you have the requirement of 3 cofactors, one is called magnesium, the other one is called S-adenosylmethionine and the third one is called ATP, whereas in the case of type 2 you require the magnesium, and in the case of type 3 you require the ATP. The product what is being generated in the case of type 1 is non-sticky ends. What is mean by the non-sticky end is that you are not going to generate these sticky fragments, whereas in the case of type 2 you are going to generate the sticky ends, and in the case of type 3 also you are going to use the non-sticky ends, which means the type 3 or type 2 restriction enzymes are actually been having the relevance applications in the molecular cloning, because of the only reason that it actually going to recognize within the restriction sites, and it actually cuts within the recognition sites and it generates the sticky ends. So, in the case of type 2 restriction sites you have the two properties. One is that it recognizes the palindromic sequences. The palindromic sequences are the recognition sites for the type 2 restriction enzyme, and what is mean by the palindromic sequence is that the palindromic sequences are, you read from the forward or the backward direction, they remain as such, because as you can see here this is one of the restriction site which is called GGA-TTT. Okay. So, in this case what you see is if I reading in this direction or if I am reading in this direction, it is actually the same, which means that if you cut the recognition sequence from the middle, it actually is going to be the mirror image of each other. You can see again, if it is CCT from this side, it is actually the CCT from this side, which means this particular thing is the mirror image of this, and that is what is called as the palindromic sequence. Non-palindromic sequence means that this is not going to follow. And in this case we have taken an example of BAMH1, which is another restriction enzyme. The second thing is that the type 2 restriction enzymes are also generating the sticky ends. what is mean by the sticky end is that if you cut a fragment, for example, in this case again we have taken a BAMH1 as an example. So, you can imagine that you have this DNA which contains this particular restriction site, and the BAMH1 cuts just after the G. So, if you cut just after the G in both the direction, what will happen is that this particular fragment will go with this DNA and this particular fragment will go with the other DNA. So, that is how you will see this is the DNA what you are going to have, which means that you are going to have a DNA like this in one side, the other one you are going to have a DNA like this, which means you are actually generating the cohesive ends, which means you are actually generating the sticky ends, and these sticky ends are going to be complementary to this one, which means if you have these two fragments and you put them back together, they will spontaneously come together and will bind to each other, because the nucleotide what you have here is going to have the complementarity to this one. This one is going to have this one. So, they will actually spontaneously will find and bind the will form the hydrogen bonding, and that has a very big advantage, because if you cut any fragment, so you can imagine that this actually belongs to the plasmid and this actually belongs to the DNA fragment or the gene, and suppose you cut the fragment with BAMH1, you cut the plasmid with the BAMH1, you are going to generate this kind of flanking sequence or the sticky ends onto the plasmid side. This is the sticky end you are going to generate on the fragment side, and once you put them together, this particular G is going to have the affinity for C and A is going to have the affinity for T, and so on. So, instantly they will come together and they will form this, and that is how you are going to insert this particular fragment into this particular plasmid, which is actually the transforming agents, and that is how you are going to generate a chimeric DNA, and then once you put this chimeric DNA into the host, you will be able to generate the genetically modified organism. Now, let me tell you how to set up the restriction reactions. So, in the case of restriction reactions, what you are required to have the DNA which you want to cut with the restriction enzymes. You can take the 1 microgram or you can take more, depending upon the amount of DNA what you want as the cutting as far as the final product. So, you can take the 100 nanograms, 200 nanograms, you can take 1 microgram, you can take 2 microgram, and so on. Then you have to take the restriction enzyme. Most of the companies what they supply is they supply the restriction enzymes at a concentration of 20,000 units per ml, and that restriction enzyme is always been supplied in 50% glycerol, so that these restriction enzymes should not be freezed when you store them in -20. So, you can add a restriction enzyme which is actually from 0.5 to 10 units per reactions. Then they also going to provide a buffer, which is actually where this particular restriction enzyme is going to give the best activity, that you have to add as 1x. So, the buffer what you are going to get is either 10x or the 100x. So, depending on what is the composition of your buffer, you can add the buffer accordingly to make it 1x. Then they will provide you the BSA, which is 100x, and that also you have to add in 1x in some of the restriction enzymes. Not in all the restriction enzymes you have to add the BSA, but in some of the restriction enzymes you have to add the BSA, and then you have to add the sterile water, which is actually going to be free of DNases and RNases. DNases and RNases are the enzyme which actually degrades the DNA or the RNA, and you know that DNase is actually cuts the DNA from the it's exonuclease. So, it actually cuts the DNA from one end of the DNA. So, if you have any kind of DNase or the RNase contamination, the DNase is going to chew up the DNA and you are not going to get the final product, because once you generate this it actually going to eat up your sticky end, and as a result of DNase contamination you are not going to have this sticky end. So, once you do not have a sticky end, this particular fragment is not going to be useful for downstream cloning reactions. Then the total volume. So, your total volume is the 50 microliter. Normally we do the restriction enzyme reactions in the 50 microliter. You will add the sterile water to make up the volume. So, the sequence in which mostly people add these reaction components are that they start with the water, then the buffer, then BSA, then restriction enzyme, and the last they will add your DNA of interest. In some of the cases what people do is they also make the master mix, which means they actually add all these components from and make up a cocktail kind of thing, and then they dispense that and then add the DNA into different reactions. Because many times what happen is that adding the 10 unit or 0.5 unit of this restriction enzyme is very difficult, because you cannot dispense 0.2 or 0.5 microliter, and in those cases people try to make the master mix of 10 reactions, 20 reactions, and then they will going to dispense eventually and then add the different DNA to set up the restriction sites or restriction reactions. One of the once you set up the restriction reactions, then you incubate these restriction reactions at 37 degrees Celsius in the water bath for 12 to 18 hours. What people do normally is that when suppose they are working with the new enzyme or working with the new particular type of DNA fragments, and they do not know how much time it will take, what they do is they start with 1 hour and they go up to the 4 hours to see what is the restriction or what is the cutting they are getting, and based on this only they actually optimize the time of cutting. The other thing what also is also important is that whether you are using the single restriction enzyme or the double restriction enzymes, because sometime it happens that you may be looking for cutting a particular DNA with the 2 restriction enzymes. In those cases the buffer what you are going to use, so most of the companies provide a table in which it is saying that if you are using the combination number 1 and 2, you have to use the buffer number 1, or if you use the combination number 1 and 4, you have to use the buffer number 2, like something like that. So, they also provide, the companies also provides a table which buffer you have to use for a certain combination of the restriction enzymes. In some cases they also say that in for example, in the case of EcoR1, so most of the enzymes, if you are doing a double digestion and you are using the EcoR1 as an enzyme, what companies recommend that you use the buffer what is being recommended for EcoR1, simply because of one point, that most of these enzymes like EcoR1, HIN3 or BAMH1, they normally also show the star activity. What is mean by the star activity? Star activity is the activity in which the enzyme actually misbehaves or it does not cut as per its recognition sequence, and sometimes the other enzymes also show the star activity because of the 2 reasons. One, that if you have the glycerol concentration, the final glycerol concentration more than 5% into the reaction. So, as I said, the enzyme what you are going to get, it will be stored in 50% glycerol. So, this 50% glycerol reactions, you have to use the enzyme in such a way that the concentration of glycerol should be less than 5%, but if it is more than 5%, then the enzymes actually start showing the star activity, which means they do not recognize the sequence of their own, but they recognize some random sequences and then they cut at the random sites. So, because of that they are not going to give you the desirable products. So, to avoid the star activity, sometimes the companies also ask you to add the BSA. The other parameter which also been contributing to the star activity is the addition of too much enzyme compared to the amount of DNA what you are adding in these reactions. So, if there is a lot of enzyme and very little DNA, then also the enzyme is actually also showing the star activity. And when you set up the restriction enzyme, the restriction reactions, you also have, you should avoid the conditions where the enzyme should show you the star activity. The other enzyme which we also having a role in the molecular cloning is the ligase. The ligase, the purpose of the ligase is to join the two DNA to generate the chimeric DNA, and it is actually been one of the essential enzyme for making the cloning possible. So, what happen is that the enzyme is having the either using the ATP or the NAD plus as a cofactor. So, in both the cases what will happen is that enzyme is generating the AMP, and that AMP is binding to the enzyme, and that is how it is making a adduct with the enzyme. And once the it is making an adduct with the enzyme, the AMP is being transferred onto the nucleotide, and then the AMP is getting incorporated, and it is actually sealing the links, or it is joining the DNA fragments. So, this is what is written here, that AMP is making an adduct with the enzyme to form the ligase AM complex. This complex is binding to the 3' end and the 5' end of the DNA, which actually has the nick, and then bringing them together. Once you bring them, the 3' and the 5' end of the DNA, they will get and form a phosphodiester linkage. Once they form the phosphodiester linkage, the AMP is going to be released, and it is going to seal the nick between the 3' and 5' end. Let us see how it is done is, suppose this is the DNA what you are putting for the ligation reaction, and what you are going to do is add the ATP and add the enzyme, which is the T4 DNA ligase, and as soon as you add that, the enzyme AMP complex is going to be formed, and that actually what it will do is the AMP will go and bring the phosphate as well as the 3' and 5' phosphate and the OH group together, and once they come together, the AMP will be removed, and you will going to have the linkage between the this nick, and that is how the ligase is actually joining the two DNA. How to set up the ligation reactions? For the ligation reactions, you are going to have the two different types of DNA. One is called as the vector DNA, which is actually could be the plasmid DNA, which you have already digested. So, both are these, the vector as well as the insert, you have the digested with a certain set of restriction enzymes. For example, you might have just digested the vector and the insert with the EcoR1 and HinD3, and the basic understanding about mixing these two is that they should be mixed in a ratio of 1:3, which means if you are taking 1 microgram of vector, you will take the 3 microgram of the insert. Then you add the T-phenotype the ligase you will put 1:10 reactions or 10 units per reactions. You will put the ligase buffer, this ligase buffer will contain the ATP. So, you have to store the ligase buffer onto the -20, so that there will be enough amount of ATP present. Sometime this ATP you have to add the ATP externally as well. If the ligation reactions are not working, then you also have to add the BSA and sterile water and the total volume. The total volume in the case of ligation reaction is always been very low, and it is 20 microliter compared to the restriction reactions, which is actually the 50 microliter. Why it is so? Because you want these molecules to interact very strongly with each other. That is why you want as lower volume as possible, and that is why you are putting the ligation reaction in a smaller volumes, because you want this OH and PO the phosphate group should come as closer as possible, and if you put it a lot of in water, the water will also come and play, will contribute into the reaction. So, you want less water, you want less volume, so that the amount of vector and insert what you are going to add will have a higher chances of interacting with each other. Then the reactions are also being set up at for 16 to 20 hours at 16 degrees Celsius. So, you can see that even the molecular dynamics is also being slowed down simply by reducing the temperature. If you remember, in the rectification reactions, we have done the incubation as 37 degrees Celsius, whereas in the case of ligation reactions, you have to do the incubation at 16 degrees Celsius, simply because you do not want a lot of movement within the reaction, so that these enzymes or the components will get enough time to seal the nicks. And then the third enzyme is the alkaline phosphatase. So, alkaline phosphatase has a very specific sequence or a specific purpose. So, what happens is that when you are actually digesting a linear plasmid containing the cohesive ends on both the side, and what will happen is that you have the phosphate on both the sides. If you have the phosphate on both the sides, what will happen is this phosphate and this phosphate will come together and will recircularize and will not let the insert to come into this. So, you can imagine that if you have a plasmid like this, and if you have the phosphate on both the side, then this plasmid itself will recircularize and it will not going to let the addition of the insert. So, you can, what you can do is you can use an alkaline phosphatase, which actually going to remove the 5 prime terminal phosphate group, and in that conditions only in the presence of insert DNA, which actually going to provide the phosphate and OH both, it is going to facilitate the ligation reaction, and that is how you are going to get the clone. This is what we are trying to say, is that if you have a plasmid which actually contains the phosphate and OH both, and suppose you put the ligation reactions, this OH and phosphate will come together and will give you the ligation product or the recircularization of the plasmids. Whereas you want the insert to be incorporated, but without having the insert, this particular thing is going to recircularize and will give you the false colonies. Whereas what you can do is, if you treat this particular product along with the alkaline phosphatase, it actually going to remove the phosphate from the plasmid, and then it will generate the OH, free OH group on both the things. If you put the ligation reaction for this, it is not going to recircularize, because you need the phosphate to form the phosphodiester linkage. So, there will be no ligation reaction. But if you insert, put the insert which will go and will be compatible with this particular type of sticky ends, and if you put the ligation reactions, this phosphate and this OH will come and will be form a nick, and then these nicks will be sealed by the ligase, and it will give you the chimeric DNA, and which you can transform into the bacteria, and that will give you the colonies. So, apart from these enzymes which are having the significant role in the molecular cloning, the people have also discovered many types of different molecules which are also having the role in the molecular cloning, and that also facilitate the insertion of the inserts into the plasmids or the transforming agents. So, let us discuss about these additional tools which people have developed to facilitate the cloning reactions. One of such molecule is called as the linker molecule. So, linker molecule is being used in those conditions when you are generating a foreign DNA, but that foreign DNA does not generate or does not have any kind of restriction sites, so that you could be able to generate the sticky ends or the cohesive ends. So, in those cases what you do is you take the linker molecules and you add those linker molecule to this foreign DNA. For example, in this case we have taken an example of decameric linker molecule which contains the restriction site for the EcoR1. So, what we have done is we added this decameric linker molecule to the foreign DNA which does not contain any restriction sites to generate the cohesive ends onto the corner. Then you put a ligation reaction along in the presence of T-fluorodenyl ligase, and that will give you a linker molecule attached on both the sides of your foreign DNA. Now, once you treat this foreign DNA with EcoR1, the EcoR1 will cut the sites onto the linker DNA, and that is how the linker DNA will generate the cohesive end. Similarly, you can cut the vector with the EcoR1 to generate the linker to the cohesive ends, and then you take this vector and this modified foreign DNA and put it into for ligation reaction, and it will give you the chimeric DNA. So, the linker molecules are a short double stranded DNA of 8 to 10 nucleotide and has restriction sites at their end, which means you have an EcoR1 site on this site, you have an EcoR1 site on this side. Okay. Linker molecules is incubated with foreign DNA and ligated by the action of T-fluorodenyl ligase to generate a chimeric DNA. The chimeric DNA is digested with the EcoR1 to generate the cohesive ends, and it is now incubated with EcoR1 digested vector in the presence of T4DNA ligase to give you the circular clone. Apart from the linker molecule, in some cases what happen is that the foreign DNA does not have the ability even to take up the linker molecules the restriction sites what you are willing to choose are also being present inside the foreign DNA, which means if you are trying to use a linker molecule for EcoR1, that EcoR1 is also present within the foreign DNA. In those kind of extreme cases, what you are supposed to do is you will use the another set of molecules which are called as the adapter molecules. So, what are these adapter molecules? The adapter molecules are the 8 to 10 nucleotide long double stranded DNA or double stranded molecule with flanking DNA sequence to provide the cohesive ends, and these cohesive ends have the free hydroxyl group to facilitate efficient ligation to the vector. The chimeric DNA containing the adapter molecule is incubated with the BAMH1 because in this case the cohesive end what you are going to generate is the BAMH1, and then if you generate and incubate along with the T4DNA ligase, it will give you the circular DNA. So, what is the adapter molecule is that it is actually the molecule of 8 to 10 base pair long 8 to 10 nucleotide long double stranded DNA with the cohesive ends or with the sticky ends already been generated. So, what you do is take these adapter molecule and put it for ligation reactions. These molecule will go and bind onto your foreign DNA, and that is what is given here. This is your foreign DNA. On one side it is adapter molecule is being formed, the other side it is another adapter molecule is formed, and that is actually the BAMH1 adapter site. And then what you do is you add this along with the and since these are containing the OH group, you also have to generate the phosphate group. So, that is why they have used the polynucleotide kinase reactions in the presence of ATP, and what it will do is it will convert the terminal OH group into the phosphate group, because these phosphodiester phosphate group onto the terminal site is required for participating into the phosphodiester linkage formation, and then you will put the ligation reaction, and once you use the ligation reaction it will give you the circular plasmids. So, in today's lecture what we have discussed about the different types of enzymes which are having the role in the molecular cloning, and then we have also discussed about the two different types of adapter molecule as well as the linker molecule which are also having the role in facilitating the cloning of foreign DNA into your vector of your choice. And what we have also discussed about the different types of restriction enzymes, their relevance in protecting the bacteria or the microorganism from the foreign DNA, and what is the utility of these restriction enzymes in molecular cloning apart from restriction enzymes. We have also discussed about the ligase as well as the alkaline phosphatase. So, with this we would like to conclude our lecture here, and in the subsequent lecture we are going to discuss about the different types of vector which you can use for molecular cloning purpose, either it is for the prokaryotic system or the eukaryotic system, and then subsequently we will discuss about how to generate the chimeric DNA and how to insert these chimeric DNA into the host of your choice. So, with this I would like to conclude our lecture here, and in the subsequent lecture we will discuss about the different types of vectors which are available for cloning purposes. Thank you.